Hi, and welcome to LabYak, where we interview some of the world's leading scientists, ask them about the science that excites them, and also ask them to take a peek into the future at what they think the science that they're excited about holds for us all. And I'm delighted today to be joined by Colin Pouton from Monash University. He's the Professor of Pharmaceutical Biology there. And Colin, welcome to LabYak.
Thanks very much, Steve.
When, just judging by your accent and judging by everything I know of having read about you, you're sat in Australia now talking to me, but you didn't start in Australia, I don't think. And I wonder if you could tell us a little bit about when you were at primary school and thinking about the future. Did science ever cross your mind? And when did you first get the bug for the kinds of things you're doing now?
Yeah, it's a good question. I think I probably really got into science at secondary school. I was at secondary, as you say, I'm originally British. I actually an unusual character because I've spent half my career, half my sort of postdoctoral career in the UK and half of it in Australia. But I grew up north of London in Hertfordshire, went to school in Hitchin, and it was really the school teachers in physical chemistry that got me interested and excited by science, I think. That took me to, I certainly had every intention of studying at tertiary level in some sort of science degree. In fact, I was thinking of doing chemistry for quite a long time, but eventually I decided to take pharmacy as my first degree. And I think it's because I had this sort of insight that I was interested in biology, although I hadn't really studied biology at school, but it must have been just wanting to apply physical chemistry to biological issues and I suppose pharmaceutical applications. So it took me into pharmacy, although in those days a pharmacy degree, I was at the University of Bath for an undergraduate degree, and the pharmacy degree in those days was very scientific, not really what it is now. So now it's very much focused on the use of drugs, the safe and clinical use of drugs, but very much a science degree in those days. So it sort of really got me into physical chemistry and the way in which that can be applied, particularly to drug delivery. And I did practice pharmacy for a short time, but I really knew I wanted to go into research. And so I did a PhD at the University of London. And it was really about that application of colloid and surfactant science in drug delivery. That's actually still what I'm best known for, actually. It's sort of lipid delivery systems to overcome the problems of poorly water-soluble drugs, traditional drugs. But soon after I did my PhD, I actually went back to Bath as a lecturer, and I was looking for what am I going to do now? Because I somehow thought that what I'd done for my PhD was sort of reached an end point. In fact, nothing could be further from the truth because it actually continued on well into the time I've been at Monash as well. But I kind of wanted to get into something new. And I think that was just a feeling I had at the time as a young lecturer. And it got me into cell biology. So actually, whilst I have still carried on working on drug delivery, I've very much devoted a lot of my research career in basic aspects of molecular and cell biology and applying those to drug delivery and drug discovery. And that's really how ultimately I came to be working on mRNA, because it's a kind of natural extension of what I was doing. I guess, nucleic acids are sort of the ultimate challenge for drug delivery scientists. And for years, I was working on trying to deliver DNA for pharmaceutical applications, and really that still remains an almost intractable problem, at least from a synthetic point of view. There are viral vectors for delivering DNA, but synthetic delivery systems really just haven't really come through. It's still sort of there on the back burner, still of interest, but as people started working on mRNA, it became much more attractive to look in that field. And obviously with COVID, it's just exploded since then.
Yeah. And do you think, I mean, when you talk about drug delivery, I mean, you're talking about inside the body, aren't you? I guess in terms of how the body works, and is cell biology a natural extension of that? I mean, as you get more and more into working out the rules of how the body works, you get smaller and smaller, don't you, in the application or the appreciation of what's happening.
You certainly do, yeah. Although I suppose in the days when I first started working on drug delivery, that really wasn't so much of a consideration. It was more to do with the sort of nuts and bolts of the absorption of drugs and their distribution in the circulation. So the sort of DMPK, as often the industry calls it, or absorption, distribution, metabolism, excretion, that was really where the focus was. But it's now very much about intracellular activity and even targeting and distribution to different organelles within the cell. Yeah, that's very much on the agenda at the moment.
And if you go back 10 years now, people talk about the last 5 years, and we take as sort of brought in the fact that we dealt with the pandemic. But if you go back 10 years, and recreate the atmosphere of what people were thinking about, was, I suppose, two things really. In the work you were doing, was pandemic or the prospect of dealing with health emergencies ever a part of it, or was it? Or was
not for me.
No, is that and as you saw the industry or you saw academia, were most people doing what you were doing, basically concentrating on really developing our knowledge and know-how, not at all driven by the fact that there were, because how could you be driven by the fact there's a health emergency around the corner?
No, exactly right. Yeah, I mean, I think locally here we have a very powerful infectious diseases institute, the Doherty Institute, and probably some of those guys who I've been working with for the last few years did have on their minds the possibility of, if you like, pandemic preparedness. But certainly as a pharmaceutical scientist, I didn't. No, it was really about looking for new applications, new therapeutic applications more than anything else.
Yeah.
But obviously, it's been a real eye-opener for everyone, really, this whole, I guess we haven't seen a pandemic within our own career time, but it's very much on the agenda now. And obviously people are looking at putting a lot more funding into research into how we can prepare for the next pandemic.
Sure.
It's certainly changed people's thinking a lot.
And if you go back then to what you were doing sort of 10 years ago, in terms of the field and sort of the footwork being done for when, and I watched an interview that you'd done on Australian television on the day that the very first injection was given in globally to someone of an mRNA vaccine. And it made me think, if you went back a couple of years prior to that, what were you doing that would be laying some of the foundations for the know-how and knowledge that was then used globally to do that?
Well, certainly we had started working on delivering RNA. And because RNA delivery systems at the moment are the lipid nanoparticles, we were working with lipid nanoparticles and alternatives to lipid nanoparticles, if you like, trying to look at what else could you use,
right, and
comparing them with lipid nanoparticles, mainly using reporter systems to look at biodistribution and translation of proteins in different tissues. I mean, interestingly, you wouldn't necessarily think that LNPs were the obvious place to go for a therapeutic application, although they were used in the siRNA product initially on Patro. They're not, the LNPs are not particularly well tolerated. Right. As I guess we all know, we've had some injected into our arms, they do have issues which the vaccinologists call reactogenicity, and it's partly to do with the RNA molecule, but it's a lot to do with the LNP itself. Okay. So we were really looking at alternatives which are, if you like, less reactogenic. But interestingly, when it came around to the vaccine, of course, the properties of the LNPs are probably ideal, really, because you do need a bit of innate activation of the immune system to actually kick off an adaptive response. So for vaccines, LNPs will work really well, but I think we still need delivery systems that are alternative for, if you like, chronic applications for therapy. So there's a sort of a whole world of people now working on alternatives, but it's not that easy to get away from LNPs. They're very effective and very very amenable to manufacturing, as we've seen in the pandemic. You know, very rapidly you could get into producing hundreds of millions of doses.
Exactly. Yes.
Quite remarkable.
Yeah.
Yeah.
Yeah. And if you then sort of roll the clock forward into where we are now, so having talked a little bit about what the challenges of using LNPs and some of the other things that you might, the systems you might use, what's exciting you most about where we could go and where we're at now and where we could go with RNA vaccines or nucleic acids, not just RNA vaccines, but how we use them?
Yeah, well, I think the vaccines, I don't suppose anyone really at the beginning of the COVID pandemic were absolutely confident that the mRNA vaccines would work as well as they did. Of course, now the thinking is quite different. It's all, people are almost thinking, oh, okay, so we can make a vaccine for anything using mRNA. Well, it isn't quite like that. Strangely, you know, COVID was probably a really easy target for mRNA vaccination because the virus is completely coated in the spike protein. Really, there's nothing much else on the surface. So if you can raise an antibody against the spike, you're doing a good job at neutralizing the virus. But obviously, all the other viral infections that you might want to vaccinate against may be much more challenging. So there's a lot of work in that area. So certainly complex viruses like HIV, you know, really they're just as intractable as they always were. Although there have been some quite recent developments with HIV vaccines, mRNA vaccines for HIV, which look very interesting. But I think, you know, from my point of view, vaccination is relatively new to me. Well, I wouldn't say it's relatively new, but it's not something that I've spent a lot of time on up until COVID. I've been more interested in therapeutic applications. So I'm sort of interested in what the next wave of applications are and, you know, finding out what we can do and what we can't do with mRNA, because I don't think we really understand at the moment how much protein you can make and in which tissues, and therefore what kind of therapeutic applications are tractable. So, you know, there are a lot of diseases where there's either a partially defective or missing protein, and you could potentially replace the missing proteins in those diseases, you know, if you could get the mRNA to the appropriate place and if you could translate enough protein. And I think some applications will require much more than others, much more protein than others, and so it's a matter of working out, you know, what will actually work. You can't make, you know, masses and masses of protein using mRNA, but you may be able to make enough for certain applications, and it's really, that's what excites me most, finding out, you know, where those opportunities are. I mean, I think, to sort of cut to the chase, I think, you know, intracellular proteins are probably the most interesting ones, things like transcription factors, where you don't necessarily need a lot of protein, and also it's really working intracellularly rather than interacting with a receptor on the surface of the cell. So, you know, then if you get the RNA into your cell of interest and you're making protein at the site of action, there's a real possibility of sort of not just providing enough, but containing the activity of the protein where you need it.
Yeah.
That's quite different to something like where traditionally we're injecting huge masses of protein and, you know, expecting that protein to be distributed widely.
Yeah.
So I think there are some sort of obvious, there are some sort of age-old targets for gene therapy which really present massive problems, like, say, Duchenne muscular dystrophy is a classic example. It's one of the most common genetic disorders, but you'd have to get mRNA to all skeletal muscle, including the diaphragm, maybe the heart tissue as well. And that's something that at the moment we don't know how to do. On the other hand, if you were saying, I can express a bit of protein in the liver, which would then have an effect after being distributed, then you have something which seems much more practically possible. We know we can deliver to the liver. If you can use the liver as a sort of protein factory, then, and you can get enough to the tissue you need to, then it becomes tractable. So, you know, that's those are interesting applications. And then the other things that are particularly interesting, I think, are the application of RNA in gene editing, where, you know, generally the thinking is that if you're going to use technologies like CRISPR and all the proteins that have come out of that Cas9 variants and so on, then you don't really want them to be hanging around in the form of DNA. So you deliver them as RNA, and they're sort of expressed locally just for a short period of time. You may then be able to actually overcome, well, you may be able to correct the genetic defect, and then the protein's gone. So there's no sort of side effect of having it lurking around for a long time. So I think those are really interesting applications, and there are allegedly some 10,000 rare diseases, which potentially could be treated using that kind of approach. So it's a huge sort of field that's opened up as a result of.
Do you think in the next 10 years you will actually see? I mean, what sort of progress do you think we'll see in that 10-year period? Because I guess I'm coming from the background of the fact that the pandemic's sort of shown us that if you actually put your energies into something, you can actually get things to happen, can't you? And I'm wondering whether that energy continues, or whether actually what happens now is we go back to the full range of issues, because that was basically by just focusing on one, as you said, relatively simple, although nobody would have thought that at the time, but we are thankful as we look back that it's a relatively simple virus that we're fighting. But the next 10 years, do you see progress in the areas that you've just described?
I do, yeah, because there are a lot of very well-funded companies around that have, a lot of venture capital has gone into applications around gene editing, for example. And also now you've got the companies that made the successful vaccines have really got some funds to play with and really start attacking some perhaps more difficult targets. So I definitely think we'll see more products coming through. I guess what's not so obvious is whether it will be quite as influential as, say, antibody development. Because one of the things is, you know, the pharma industry, if you look back at the time when I did my PhD, the big pharma industry really didn't have much interest in biologics at all. But now, of course, it's almost dependent on them. And the biggest pharmaceutical products of all are biological agents, generally antibodies. So, you know, that if you look at the antibody field, it's monoclonal antibodies were first described in 1975, I think. And then it took maybe 20 years before the first product came through, but it's just started an absolute avalanche of therapeutic antibodies. And of course, the most profitable products of all are antibodies, but you've got 100 approved antibodies, 100 FDA approved antibodies now, and another 500 or 600 in the clinic. So it's just extraordinary acceleration. And I guess what's not obvious is whether mRNA will take a course like that and have, you know, quite such a massive influence on the industry.
No, I think that's right.
It could well do, but because we don't know.
No. How easy do you find it to attract people to join you in your research then? Is it the kind of stuff that is inspiring young people to come into the industry? The story of the last few years, has it made any difference at all?
Yeah, I think it has. We're a bit, things are a little different in Australia to Europe and the US because we have an exceptionally strong scientific base here, but we don't have much of a pharmaceutical industry.
No.
It hasn't really stopped us working with industry because we work internationally, but there aren't so many obvious jobs for graduates to go into from, you know, an academic study here locally. But it didn't seem to stop them wanting to study. We have a lot of very good students, often from our own faculty, actually coming through, and they tend to go off into academic research or sometimes into jobs that are associated with industry, but not necessarily R&D jobs in industry.
Right.
And some of them do go internationally, but obviously at the moment in Australia, what we're really interested in locally is building an ecosystem around this mRNA space, which can generate jobs and careers for a lot of our young scientists.
Sure.
And that doesn't mean to say we don't want them to go overseas, but
No.
there's an opportunity now to develop an industry, particularly around this mRNA space. And actually, the local governments in Australia have really invested quite heavily in promoting mRNA research. So it's an exciting period for the people in academic institutes.
Okay. No, that's fantastic. And just finally, as you do what you do, how do you relax from what you're doing? Are you someone who works 24/7, or have you got other interests in life?
You might get a different answer if you ask my wife on that particular question, but I don't really. I really enjoy doing research, and so I wouldn't say I don't feel pressurized now because I'm too old to be in that
Right.
state of mind. But I just enjoy it. But I'm not really 24/7 anymore. But and I don't feel I'm not sort of in the same state that I need to work really hard to get the next promotion or the next fellowship, that sort of thing. So I'm enjoying my research career at the moment. And you know, I haven't really ever had any problem with motivation, actually. I'm sort of lucky from that point of view. But I think it's because I've never really restricted myself to a little small area. I've got a sort of very broad research portfolio, I guess. And it's probably the result of never really having a supervisor when I did my PhD. Basically, I had a supervisor for 6 months, and he got a job in the US. And basically, I just was on my own. And so it was a slow start from the point of view of publishing,
Right.
going to conferences and that sort of thing. But it basically, I sort of learned to do stuff and had no fear. And so it made me tend to enter fields where perhaps I shouldn't. But
Wiser
from that
counsels
point
might
of view,
have
it's
told you
kept
not
me.
to. Hey, well, Colin, it's been a real pleasure talking to you, and you've painted a really positive picture about where the next 5 or 10 years might go with the mRNA technology, and also what the science that you're excited about, the part it's going to play. Thank you so much for your time, and thanks for being on LabYak.
That's a pleasure, Steve.